worse. Thanks. Okay. So today we begin. So far, we have mostly discussed. You're not on the photo. Oh, I'll get to that in a minute. So far, we have mostly discussed about what I would call the background and how you can infer function by recognizing some similarity to existing proteins. And today marks the beginning of somehow a new chapter in function prediction, where we talk more about methods that really predict function from the get-go. And the first topic, major topic, that will begin today but will extend over the entire next week, most likely, no, more than the entire week, is localization. So again, the background here, and let's have that off, the prediction of subset localization. So let's begin with the terminology. By homology, I explained. So the idea that to infer any aspect of function by homology means ultimately back to the roots, the evolutionary relation. The assumption is that we can infer similarity in function from similarity in sequence because proteins are related. They evolved from common ancestors, and that's in the root of the term homolog. Then there are two different terms, de novo and ab initio, both Latin. Does anybody have an idea what they may mean in terms of prediction? I understand that you haven't seen those terms before. There are several people here in the room who have an origin in a Latin language, so they immediately understand what the meaning of the word is. But in English, this is initial novel, so this is the same root as the Latin root, also the English word. But what could be the difference in terms of predicting something? Any idea? Any guess? No idea? So ab initio, from the initial, somehow goes back to the idea of physics. You write down an equation. There's nothing you know except for how a system works, how the ball flies, Newton's equations. So this is something that in principle you can walk to your room, think about it, and write down an equation, and that may describe nature, right? You simply think or observe and then think about the first principles. And that explains this. So this is like molecular dynamics. When you, in fact, simulate protein structure formation through the equations in physics. De novo, the idea here is you use some information. That information could be machine learned. That could be data mining. So here you use the existing knowledge to somehow extract something that will allow you to predict for new proteins, for proteins that you have not seen before. When you make the inference, you do not need a similarity to something that exists. When you build up your model, then you need things for which you know, and you need sequence similarities. So essentially you start with a bunch of assignments, for instance, where you by homology see something, and you hope that you extract the principle here without actually in your search having the homolog. Right? Is that clear, the distinction? Not so clear. Again, by homology means that you start with a protein for which you don't know what it does. You look up in a database of things for which you do know what they do. And you find a sequence similar one. That's homology. You assume there's an evolutionary connection and that you can infer function because of this historical connection. Is that statement clear to everybody? Yeah. Okay. De novo simply means you machine learn, you train some feature. You extract some common feature, some motif. Okay? And from that motif, the next time you predict, you just ask yourself, is that motif existing or not? You're no longer looking into a database. Okay? It may be that that motif relates, and I'm going to show you cases where that motif directly relates. It's not really machine learning or the simplest way of motif extraction. That motif relates directly to a protein where you can say, okay, you call this nuclear because the motif is in this protein there. Okay? And I understand the mechanism why you call this nuclear. In some cases, there may be a little bit of this, a little bit of that from different proteins sort of put together, right? It's a sort of generic motif that the machine learning device learned. Okay, the problem, again, this is Mycoplasma genitalium, and the stress is on two aspects, or various aspects here. One aspect is there's a densely, this is the cell, and it's completely densely packed. Again, it's good cell, illustration from good cell. This is the lipid bilayer that protects the cell against the environment. Now, simple prokaryotic cells, prokaryote means karyot is the Greek word for nucleus. The core, pro is before organisms develop the core. So organisms without a core, that means. And essentially you see pieces of DNA around here. You see a cell wall. In some bacteria you have this flagellum. You see ribosomes, energy factories. But ultimately all of this swims around in a so-called, we could call it soup. But it's not soup. Remember this figure that I showed you before, highly packed. So a very dense borscht or something like, or no, even that is right. So a soup with not a lot of liquid. Okay, now in this particular case, we could still imagine what we want to predict is the difference between things that are somehow in this white, that are in this yellow, and that are outside. Why would an organism send proteins outside? An organism, a prokaryote. So I'm talking about, in this particular case, in the simplest version of a localization prediction method, where the protein is, we would have something inside in the white here, something in the yellow, and something outside. Now the question is, in this three-state model, why do prokaryotes have proteins, genomic material outside? Or not genomic material, but proteins outside. Do they? So this is the wall that protects them against the environment. Inside is self, inside is its life. And most prokaryotes are actually single cell organisms. So it's just this thing that is the organism. So why do they have things outside? Why do they send proteins outside?
To react to stimuli.
To
react to stimuli, to interact with the environment.
Those are two different things. So react, or is it the same?
I would say reaction to stimuli is a subset of interaction with the environment.
Okay, so there's an interaction with the environment, you say. What could be the interaction with the environment? What could you...
Rejecting carbon dioxide, for example?
Yes, you could, for instance, sense that there is a high... So you have some food source, and you sense the direction of the food source, and that's the direction in which you move. Okay, that's what the flagellum, for instance, is used for, to move yourself around. You could sense an attack, and you could sort of mount a counterattack. The bacteria don't have what we commonly see as an immune system, but a protective system they do have. And you could, mostly this is sort of driven by food sources, but in fact some of these bacteria live in colonies. So maybe you don't sense the food source, but you sense that other bacteria move in some direction, and you believe that's the food source. So there's a variety of things that happen in the communication. So the next step, yeah?
I have a question, it's slightly off topic, but you said that reaction and interaction with the environment are two different things.
You said not. I took your argument.
Because I'm trying to think of interaction that is not driven by a reaction.
No, I took your argument. So that, in some sense, you may call the step up in evolution, the evolution from prokaryotes to eukaryotes. And eukaryotes exactly have the core. What's the most common feature of a nucleus? What do you have in the nucleus? Do you know that? The DNA. So the entire, now we get a little bit complicated in my definition, the entire nuclear DNA is in the DNA. This is a little bit of a circular definition, but typically what we call the DNA of an organism is in the nucleus. Do you know any other DNA that is? The mitochondrial DNA, yes, that is swimming around. In the mitochondrial DNA, there's relatively little. So there are many mitochondria. So most of the genomic material in a cell are in the mitochondria in people, but every single package, so there are hundreds of mitochondria, but every single package is a few hundred in the 100 megabase, while the nucleus is in the gigabase. So it's many orders of magnitude higher, 2 orders of magnitude higher. But again, there are hundreds of those. That's more than 2 orders of magnitude, and that sort of equals out the numbers. There are other compartments. Now, let's get back to the nucleus for simplicity. Why would you invent the idea of a nucleus? Why could that make sense?
Protect.
essentially is a protection mechanism, right? That's one aspect. The other aspect, so protection against, in some sense, protection against, let's call it foreign influence, but it's also a protection. So this is where if a virus wants to attack or any intruder wants to attack, they want to put their DNA to be replicated, right? And somehow it's a step removed from the cell wall, a step removed from the attack in that sense. But there's another aspect. So the other aspect is when you put a shell around, in this particular case, the DNA, you can have this environment have a different biophysical environment. So DNA is very positively charged, so you can sort of keep your positive charge in one room. Like you have a fridge, like you have a cold room or something like that. Maybe the analogy with cold is really wrong here, but you have different conditions. In a heated day, you have one place where you have a different temperature environment. That also means that you can put everything that sort of works on DNA in that compartment. So you have a compatibilization relation helps you to in fact have specific purposes dedicated to certain regions. Put a membrane around, you protect them. Or you make sure that nothing interacts with this DNA that is not entering through these membrane. And this essentially is the same for the other compartments too. In a very simplified view of our prediction task, we only have a nucleus, we have this thing in between, and we have the extracellular space. If we want to predict it, again, the most common way of doing that, and here I prove myself wrong, I'm going to go back to homology quickly, but most of that I already showed, is by sequence similarity. So we are back into the homology idea. There's text similarity, and I will talk about that today. And then there are motifs, and then there's sort of really de novo prediction. This is as far as I will get today. So the de novo prediction I will begin to talk about, and I will continue next week. So again, in order to set up the homology-based inference, I already said we build a data set of proteins of known localization. We take a unique subset without bias out there. We align this against all the others, and we simply define a threshold in blue here according to some level of sequence identity at which we can infer localization at whatever threshold we want. We cannot really get 100%, but if we wanted to achieve 95%, then the threshold would be somewhere here, and so forth and so on. That's the blue. We have talked about that. So, let's talk about text analysis. And the work is from Rajesh Nair, who is now at the FDA, the Federal Drug Agency in Washington. And he got into this work through a review on a paper that he wrote. Now, I'm not entirely sure how often that will happen in your life, that you will get reviews on manuscripts that you write, and that you will have the sensation that the reviewer did not quite understand what you wrote to the level that you understood it. This may also happen with your supervisor, that may happen with your friends, may happen with all kinds of people around you. Whenever this happens, see that as a positive challenge. Usually you are so much in your topic that you no longer sort of see the connections. Very often different views help you to get out of local traps, let's call it like that. Any interaction, even if it is very, very negative, see the positive spin in it and see to improve, grow with it, right? So the most extreme example is a manuscript that Rajesh sent in which he predicted subcellular localization using neural networks that used no information. So there was a de novo prediction method. And one of the reviewers said, so he had 3 reviews. One was very positive, one was neutral, and one was very negative. The very negative one said, there are much better methods out there for doing text analysis. Now, text analysis was not mentioned in that paper, nowhere. The paper had absolutely nothing to do with text mining, text analysis. And the review went on how the wrong measures had been used, but the measures had not been used. So possibly the interpretation was this reviewer had reviewed a different manuscript. The alternative way is that the reviewer was really focused on giving out a message of what that person wanted to see. Both could be true. Now, most people would have said nonsense, right? Rajesh went ahead and said, oh, if that's what they want to read, I'm going to send in a manuscript that has text mining in it and predicts localization based on text mining. And that's what I'm going to present. He won an award with that. This is a completely unique story. He really did that. I'm not making up any part of it. Just imagine yourself, sit in a quiet corner and imagine yourself reacting like that. Very few of us would do that. This is the right reaction. Learn from it. And I've been trying over the last 15 years to copy Rajesh. Back again to the origin of the data. This is a particular SwissProt record. So SwissProt is the database of annotated proteins. It is created by people reading the literature. So somebody, an expert in biochemistry, reads the literature and sees in the literature that this particular sequence that is not shown here, that's underneath somewhere, is in fact claimed by some paper to be transcription factor. This is the name, this is the description found, that's the species, taxonomy, so in what kind of tree it comes from, part of vertebrates and blah, blah, blah. And then descriptions of function, they typically, and again there's a column down there where it's explained where citations of these statements are given. Again, every single record is written or is annotated by an expert. And many of them are annotated by many experts. So most of these records do not stem from one publication, but from several. So you could imagine that, you know, it binds DNA is a paper, and then there are more details revealed over time, and every single time this record is updated. Of these, SwissProt at this point has about 400,000. To get there are tens of annotators who work every day non-stop on this. Now, one particular column here says it is nuclear. If you look here at one of the issues that we already discussed, and the nucleus is where the DNA is, right? We already also discussed that the only other part in the cell where you find DNA, in the eukaryotic cell where you find DNA, is in the mitochondria. So binds DNA. If you just read this statement and knew this, then you would immediately know this must be nuclear or mitochondria, provided this is the correct information. And the idea here, if I didn't know that it's nuclear, if I didn't know that binding DNA means nuclear or mitochondria, because I need expert knowledge for that, could I not just take these keywords and infer the subcellular localization from those keywords? How would you do that? Well, let me just get, before we get to answer my question, into the next. So there are two aspects actually. The one is what I talked about. But in these particular cases here, you, just like the one before, expert knowledge gives you the annotation. But the challenge would be also to, in fact, pick up annotations that are not trivial, even if when you are an expert. So you put an expert in front of that list, and they still wouldn't be able to say, okay, that's nuclear. DNA is simple. Chromatin regulator, blood coagulation, all of these are simple. But there are some that are not simple, and I'm going to show you some later. So we have two goals here. We want to find what is trivial for experts, but we want a machine to find it. And two, we want to define things that are non-trivial. How could we do that? Any idea? So I give you a bunch, and let's just begin with the data. I give you a bunch of proteins for which I have annotations, these keywords here, for particular subcellular localizations. So 2,900 have annotations in their extracellular, 1,700 are mitochondrial, cytoplasmic 2,600, nuclear about almost 3,500, chloroplast, only plants, 1,600. And here are some of these annotations that I find. But how could I develop a method?
Try to cluster the information you have.
How would you do that?
Well, it's labeled data, right?
Yes.
So we, I don't know, try to cluster it, find a correlation between certain keywords.
Yes, how would you do that?
Well, I don't know, first discard articles and pronouns and all that.
That's the first step.
Meaningless? Yeah.
So, but how would you do that actually? I mean, one way of doing that is you use a text mining tool, right? That in fact tags everything that is the A and blah 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 blah. But there is a much simpler way to do this.
Lookup table of expressions you want to look for?
That again would require that you go in with an idea what you want, right? So Dimitri is totally right. This is a simple way of doing that, but it requires some prior knowledge. The knowledge what is A? What are meaningless words? Can we not? So initially you said cluster, data mining clustering. Could we not sort of make the cluster sort it out itself? It's a very, very simple thing, a simple way in which you can do that. Anybody an idea? Well, if you have things that are meaningless, like the, most likely you find them in all classes. Right?
But what you don't find in all the classes are the order. I mean, the D, A, B in that order. That's, I mean, it's a segment what you don't find.
You don't find what? I'm sorry, I didn't get you.
I mean, you don't find the units because it's only just 4 letters or 5 letters.
Oh, words such as A, B, C? Oh, no.
No, you find like a combination of them in all the extracellular, all the cytoplasmatic. They have a signal, all of them, or not?
A sequence signal, or what? I'm completely confused. Are you asking me a question? Or are you telling me something?
I'm asking a question.
Is there a sequence signal? Are you asking?
Is there a sequence segment that will?
But we're not going to talk about that. Yes, that will be the next step. There is, but this is not this method. In a few minutes, or in 10 minutes, I'm going to talk about another method that uses sequence segment, so to speak. But this one doesn't. This just wants to classify the keywords such as, so we are not using sequences at all here. We are essentially using annotations. Glutamate biosynthase or something like that, or proline biosynthesis, or DNA binding, or blah, 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 blah. So again, how could we do it? I mean, I believe essentially I already outlined one idea how to get rid of the.
Well, yes, you said cluster information, and if a certain term is in every class, of resulting classification, it means it's meaningless and we can simply discard it.
You simply compute the Shannon information, so the information content of a particular keyword for this problem. You have a final goal to use the keywords and use these keywords to sort it into those classes. That's a clear objective. For a clear objective, you can clearly measure the information content for each keyword. You can throw out things that are not informative like the, and you can keep things that are more informative. So ultimately, that leads to a situation where you will take all these proteins that you have, 15K, extract all the keywords, and then simply classify and then build. The simplest way then is to build a vector, where when you throw out things that are not informative, you keep everything that is informative. Now we get into the assignment problem. You could just, again, look up the cluster and look at the probability. But here's a sort of, in some sense, more advanced and in another sense, simpler way of doing it. So each row here is one protein. The columns are the keywords. In this particular case, I show 6 keywords. They happen to be grouped by nuclear, extracellular, cytoplasmic. And for every single protein, you simply look up in this keyword vector, is it there or not? Okay? 1 or 0. It's a completely binary vector. How do we proceed?
After having this database. So we have the database, it means we got all the data we need to make a decision into this nice serialized form. So now we take a new protein, get its annotation, and then try to score it.
How? Yes?
You can create the decision tree.
Yes, you could do that. Actually, that would be an interesting idea because it could possibly be very fast. The problem is that in the tree you make you assume something about the structure of those words. You assume something about a priority. You could do that by, if you had a very good clustering by information content, then I believe you would have a good hashing tree built into that right there. But the problem is that if your first keyword was wrong, you can never recover from that mistake. So that would be tremendously fast. And in cases where all the keywords are completely right, would be a good idea, I believe. But if a keyword were wrong, that may not be the best idea. Yeah?
Because we built a decision tree from this database.
That's
and then
no, but that was this idea. I believe I shot it down.
No. And then I mean use it not a random a forest of decision trees with a certain degree of randomization. Wow, that would eliminate a mistake of a certain.
Yes, but I also would eliminate the advantage of the tree. It's true what you say.
But
yes, you countered my argument, but you killed also the motivation for the tree. So yeah,
A decision tree.
A decision tree in this particular case. So let's get back to the example of the enzyme classification. So at the root, you would say it's an enzyme. For enzymes, I can have all of these, and then you go further down in the tree. You simply narrow down the options that you have. The further down the tree you go, the more toward the detail you go. So that would work if you had some keywords that are particularly high level. Calcium binding would be an example for that. Calcium binding you can have everywhere, so that could be sort of possibly leaning toward more than one, more in one direction, but not quite everywhere. So you reduce the options.
Okay.
But it means you have some inbuilt hierarchy. And again, you could build, so I was a little bit fast here in my explanation. So the inbuilt hierarchy would be the case if you had keywords that are really classified according to information content. If you information content clustered your keywords, and so that's high information content, whatever function you really have. It could even be totally flat, and that is low, and this one here is count. So even a flat distribution here could be such that you would say, okay, all of those with a very high would be in the top of the tree. I would make the first choice. So my first statement here was that we are not completely able to rely on our information content classification. We can classify things that belong everywhere, like that's easy. But then to sort from informative to more or less is complicated. My second argument was that in fact, if in particular whatever is high in the tree, say I could do high, if I could do that, if whatever I put on top of the tree here, if that keyword is by mistake, and this keyword makes you go in this direction, and then you go further down, and maybe many keywords down here are right, but they belong to a different part of the tree. You made a mistake already here, and you will never be able to recover from that. That's why Dimitri argued, okay, let's not do a tree, let's just have a forest, so many trees, where in fact I sort of go down alternative trees and see where I get. My argument against that was then you lose the advantage. So the advantage of a decision tree is very fast. So every elimination here really reduces the CPU time, rather than in this particular case I will tell you we have vectors of the length of 400. In a decision tree, essentially you have a tree built on 400, so that maybe 5 or whatever the number is. You may have only to pass 5 decisions. This is really fast to compute, yeah?
Professor Rost, but having a forest linear in the number of trees would still be linear in terms of computation time.
Yes, but so it kills the argument, because you would possibly go here into 100 trees, right? At the moment you go into 100 trees, you can as well compute the 400 vectors. So the big, again, it would work, I take your point, it would work if you, I'm not sure what you call a tiny forest, with 5 trees or something like that. There's a particular German hunter word for that, but I don't know the English. Yeah?
Could we build a profile after getting this cluster?
Yeah. What do you mean by profile?
Well, like we did with sequence families, with conserved regions.
Yes, we could. But look, the solution is already here.
Well, we can compress them in a single profile.
Oh, okay, let's not improve. Let's first... Okay, bear with me. Bear with me. We get back to that. We get back to that idea. But for the time being, I begin this way. I'm asking you, what will be the next slide? How do I continue?
I continue by assigning the same score for our new sequence.
Yes.
I presented it in the same way.
How do you compare them?
Huh?
And how do you compare them then?
How do I what?
Compare them. So you have a database with these foreign component vectors. So the components 1 to 400, and then you have a new with the components 1 to 400. So what do you compute?
Scalar product.
That's a simple way of doing it. So we have the experimental database. You start with a new vector. You assign the vector. Here's another issue. Since we have a vector, we in fact have to assign subvectors to it. And that essentially is if a keynote would be wrong or missing. So we have to look for a bunch of other vectors that could be taken with those words, but just leaving one out. Okay? So I create, you don't have to do that. Well, you're doing safer in the recovery. Yeah, that depends on the product you do. So not doing that did not give a similar entropy-based classification. In this particular case, we did not quite use the regular product. We just used the Shannon information, which in principle is down to a vector product and then computing the story better. Now, that works to some extent. So with a query fitting into the database, it finds an assortment, since we have a Shannon information to compute from that product the information content. We have one hit. So again, we want to classify into nucleus, cytoplasmic, extracellular. In this particular case, more classes than that. But we will simply take the hit with the highest information content, whichever that will be. Okay? But of course, there is a second best hit. And in our particular case, the database was larger. We did not only have 3, we had 6 or 7 classes. So
I have one question. Why do you match only the ones instead of 1 and 0?
no,
Like the columns are only for the ones.
I don't see what you mean. I have zeros. Maybe you cannot see it. No, that's. I'm sorry. This is visual.
Okay.
No. We really. Oh, no, I get your question. I'm sorry. So we really compute this entire Shannon information. We compute the ones and zeros. The zeros have just 0 entry here. So the effective contribution is the one from the ones. Visual guidance. So back again to the second hit. So you can ask yourself, how well do you do if you look not at the best hit, but the first 2. What I show here is accuracy or precision versus coverage recall. The first hit and the second hit, the top 2 hits. So it goes clearly up, right? So at any level of accuracy, well, at some point there's no real difference. This is partially resolution of the data, so we don't have enough data to really make a nice plot here. Most likely the plot is going to be something like this here, and the deviation from the shape like this is the mistake in the data set because it's too small, is my expectation. So here you have the largest deviation because here we have very few counts. But when we get to this point here, the point is, for instance, 87% accuracy. Taking the best hit, you recover 25%. Taking the best 2, recover 30%. Conclusion, taking the first 2 hits is the better strategy. Is that right?
I don't understand how we decide between the best 2.
The best 2, again, I have a Shannon entropy, so I have 8 different classes. In my database of known keywords, I've assorted keywords by 8 classes, and I have a hit, a Shannon entropy with my query against each one of these 8. And one comes up top, one comes up second. So I can compute how well do I do if I take the top hit. That's the blue. I can also ask, well, if my true one was not the top, was it at least the second best? Right? And that question is answered in the red. Since the red is considerably higher than the blue, the second still contains a lot of important information for the prediction task. In fact, in many cases in which the first one was not right, the second one was. Now you could ask how does that play on for the third? That is not my question here. The question really is that enough for deciding, okay, from now on I'm going to recommend to the user that in fact using the first two is the right strategy? Well, okay, the user may come back to you and say, no, I only want one. But it's not the question that I'm asking. So clearly, if you have a user who you could ask, you have to ask whether they could take two. But before you do that, the question really is that a better strategy? So these numbers are higher, yes, no doubt. What you clearly now will have to ask me is what are the standard deviations here? What's the error bar? Before you can answer it. Okay, I'm not giving you the error bars, but this is above the error bar. Okay, so what else do you have to look at in order to answer my question? When you see, again, this is something totally simple, and you will encounter that in your work. There's another question you have to ask in order to advance and say, well, taking the top two is better than taking the top. Again, it is better because red is higher than blue. But you also take two instead of one. So how can you sort of see the balance between these two effects? What do you have to do?
Or maybe you can apply, like, keep the best two and apply some other technique that would be more precise.
Yes, that is a. We get back to that. We get back to that. This is in fact, we get back to that because it's a very central issue for machine learning. We also get back to your other method story, but this is not what I mean. Much simpler. And
only the second is, that would be interesting for me at least.
What do you expect?
Yeah, there are two ways. The first one would be that it's still better than the blue curve.
That would be bad.
The messes aren't really bad. And,
we would actually, in some sense, we would jump back to this one here. Resort and always take the second, right? Yes? Or it falls below. Then what do you learn from it?
And then I wouldn't have learned too much because that would be natural that if I just increase my bit size, then I get more of it.
True. This is an interesting idea. And it may, in fact, the reason why I like this idea is it is the only way in which you can find out whether your method is really messing up completely something. But there's something before that. And that, in fact, answers this question in some sense too. And that you always should do. So the question ultimately is this gain simply coming from the fact that you allow yourself two instead of one pick? You have a bunch of possibilities, in this particular case 7, I believe, and you randomly pick. And that is a random throw. And you have to see whether two random picks, how much is the gain of a random of the blue? How much is the gain of a random for the red? So for two picks, you have to compare it to random. And if, in fact, the gain is not much more than what you get out from the random, you immediately know where the second alone would fall. Provided that the first is clearly much better than random. In this particular case, that was in fact exactly the case. The first was much better than random. The second barely improved over random. It did improve over random, but not importantly. So plotting the second alone would have been substantially below this. Which in fact, had you done that, so I like your idea, but would you have done that, you still wouldn't know about the random thing. And the random thing you always have to do. You always have to put whatever number you claim in terms of a random background. What could you have done? So random is one way of putting it as a background. Something else is existing methods or some sort of, let's call it clever random, where you know something about the problem. Let's not get into that problem at the moment. Let's get back to, by the way, there is a relation that has to do with a different localization, slightly different for different classes. But in what I said, I will show you some examples of keywords that in fact are not trivial at all. All of these are not trivial, and they work only in combination with other keywords. None of them leads you to correctly classify the subcellular localization, but in connection with other keywords, it does. So let's get back to the first statement here, a serial statement. Let's try another technique. So the idea is, let me rephrase that. I'm not getting back to you, Cyril, yet. I'm still misphrasing what you said and try to illustrate something else. The something else is the following. I have method A and I have method B. And I know method A to predict subcellular localization in 3 classes, say gets 70%, and this gets 65%. So A is clearly better than B. And, okay. Now we have cases where A and B agree. Okay? What do you believe for cases where A and B agree? Are they likely to, for the subset of cases, say this is based on 1,000 cases, the whole thing here, and say for 800 of these cases, A and B agree. A equals B for those 800. What do you believe is the prediction accuracy for those 800? Any number. The number is not really important, but I'm essentially asking, will it be 65? Will it be 70? Or will it be higher? Most likely. Again, you like to say we cannot make a statement because it may be different. It's absolutely true here. But what's the most likely answer? Above 70. That's typically the right answer. Now, that induces people to, rather than taking method A or B, take the subset where they agree. Is that the right strategy? Do we let's put it differently. From whatever I put on the whiteboard, have I established that, because say this is 72, above 70, so say it's 72. The cases where A and B agree. Does that mean that it's the right strategy to always look for the agreement between 2 methods? What is missing? You're right, it's not the right. Exactly. That's exactly the point. So in a plot where you have accuracy versus coverage, or precision versus recall, for very high accuracy I have typically low coverage, and then typically I have a curve like this. And say for some level here, the number in fact is 70 for method A. And say this is the curve for method A. Now this is 80% of the data set. And maybe for 80% of the data set, in fact, method A has 80%. So one way to get at 80% of the data set is by combining wherever A and B are the same. Another one is I let method A alone decide what are its 80% top predictions. And in this particular case here, okay, I have to use different numbers, otherwise the 8 is confusing. So in this particular case, say the value that I look up here at 80% coverage, so that's the, okay, my plot are really not good. What am I doing wrong here? I redo my plot. Coverage, accuracy, at 100% coverage, I'm here. I get 70% accuracy. At 80% coverage, I get 77. So it's more like this. My plot is more like this. Anyway, did everybody follow me? Despite the complication with my graph, suffering in the group mounts. The point is that by combining methods, implicitly you reduce the number of cases you look at, okay? And in this ROC curve, or accuracy versus coverage curve, you have to see whether the jump or the reduction in coverage leads to the same or higher increase in accuracy. For well-assessed methods, combining two methods typically is not as good, okay? Back again to that issue. Now, say for 80% of the cases, A and B agree. Now, the idea could be for the 20% where they don't. Okay, by the assumption of my case here, A is better for those. We already know we should better take A. Anyway, by my construction. But I misinterpret Cyril's idea to mean that why don't we build a prediction method for those cases where they don't agree? Or in a more generic question, whenever my method A goes wrong, why don't I look at the results where A goes wrong and develop a method for whenever A goes wrong? So, what is your reaction to that?
But you don't know when A goes wrong.
Yeah, you don't know when A goes wrong, that's correct. But you can look at it, right? You can look at some cases. What can you see? So for instance, you could see A goes always wrong when the word "the" is not in the database. What would you learn from that? I would call that a lucky hit. What would that imply? Guys, you're computer people. You're programmers. What does it mean?
Fix it.
Yeah, that's a bug. Whatever you call it. I mean, this is a clear case. Programming mistake. You have some wrong if loop somewhere. Something went wrong along the lines, right? That cannot be. This is a systematic mistake. Nothing in what we discussed here, the didn't even show up. So if the program is sensitive to the, then there's some programming mistake somewhere. Okay? That's very clear, and that's simple because it immediately is helpful to correct something. But let's assume it's not that trivial. Then what people often do is they see certain number of cases. So they see there's a certain probability that the ones where they go wrong, they are nuclear. So I do worse for nuclear. Well, I can now learn a separate method, C, that is nuclear only. Okay? And then I could, for instance, compare the generic or generalist A, or let's call it the AN, and the AN, so the nuclear expert. Okay? The problem that we get in this case is, how do we combine them? And that problem would have to be addressed separately. Ultimately, in some sense, the problem of this approach here is that we believe sometimes we can see signals from cases we look at and they are not real. Ultimately, the only way in which I can improve is raise this level. Develop a new method that gets a better level. That's the only safe way in doing that, from the get-go, after I fixed all my programming mistakes. And that is back to Cyril's idea. What did you mean by other technique?
So once we have the top two hits, we want to compare them more specifically with each of the two hits. And for example, we can use maybe word co-occurrence matrices to see if maybe...
just those two?
Yeah, that's what I had in mind.
But that's interesting. So that's a very interesting idea, because you would, in some sense, trace a higher order correlation. That higher order correlation. The only problem with that, well, again, so the only danger. This is a great idea. Didn't occur to me. The only problem with the higher order correlation is typically you need a larger data set. Our data set was okay for what we did. So we had 10,000, we had a vector of 400, 10,000 cases or 15,000 cases. This is comfortable. And in fact, it may even be 400 times 400 gets into a number that is a little bit too large for, or at least for all cross correlations. For some, it possibly may. Interesting idea. Never had it. That's a great idea. But again, I also remember in the answer, when you have an idea like that, think about how your data is supporting that and what this particular idea adds in terms of complexity of the problem. So just comparing pairs in terms of the way the algorithm works is a square phenomenon. So essentially, okay, I could sort of, again, I said I could cluster it, but essentially by looking at two keywords, we have a vector of 400 components. By looking at two keywords, essentially we do a 400 times 400. So we blow up the free parameter space in some sense. Okay. Now back to Dimitri's point of other method. You said alignment.
Yes, it's creating profiles from our clustered classes. We have one set, zeros, positive hits and negative hits. Could we compress that information into a profile, create a profile for each class, and then... now I get it. So you have taken too many of my courses. It's officially the time to leave it. See you. You learn too much from biology. In biology, these profiles are immensely powerful. So you have the sequence 1, sequence 2, 3, 4, 5, and where they align, this tells you a lot about the function. Now, here the idea is to, essentially, this is like a vector. I have a sequence that is a vector. And in this particular case, I would then have the sequence 1 would have components 1 to n, so its length, and then in this particular case I have 4 vectors. And Dimitri simply puts the analogy and says, okay, it's the same thing here. I have vectors of length 400, so these vectors are length of sequence, those vectors are length of keywords, but I have the same thing compiling the profile. Here, what I know is that running sequence 1 against 2, so just doing pairwise sequence alignment, is way less accurate than running a sequence against a profile. So the averages in each column. The averages contain a lot of information. So why not do the same thing here? Anybody sees the answer, why not? So here, the information of the sequence is, and now really the problem is, this is really evolution that makes this so valuable. Any stretch of K here, of like 3 mers, is relevant only because it evolved this way. In the evolution, those 3 were essential, and because those 3 were there, you could change something or not change it, depending on what you see. So the letter I and I plus 1 are highly correlated as an or I plus 2. So consecutive residues are under similar evolutionary pressures. In the vector of keywords, I have absolutely no pressure, I have absolutely no reality. As a random assignment, there is no order, there is no truth, there's no knowledge in these vectors. So that in fact is also the reason. Try to find correlation between those keywords.
Yeah, that's again what Sybil said, right? This is essentially looking. I mean, he said this, made this point about this when looking at the second one, but ultimately this is exactly the pair correlation. So we could look at all the correlations between these keywords, and in fact, I do believe today we could actually do that. So the fact really is that I never had the idea. So Rajesh published that, and we were happy with this, and we never got around to doing this in a different setting. We began doing it in a different setting, but never came far. But the pair correlation is a really interesting idea, which possibly today we would have enough data. True. Okay, anything else so far about keywords? No? Okay, so now we get into the theme that you mentioned, Muriel. The theme of motifs. So motifs are of two types. There are two types. The one is, in this particular case, called signaling. In this particular case, it is terminal, but that is not really the point. I should maybe change the way I plot them. What I mean here is you have a sequence consecutive, long theme, a long sequence of residues, sequence tag, whatever you call that, that is responsible for the subcellular localization, that is recognized. It's like a zip code. Okay? In the other case, the zip code is distributed along the sequence. Okay, you still have a zip code, but you don't see the zip code before it forms a three-dimensional structure. So you could imagine that in fact these are, what you need to have here is a cluster of positively charged residues. Okay, so you have a positive charged residue here, the positive charged residue here, here, and here. On the sequence alone, it may not be anything interesting, but once you bring them close in space, okay, what, you want to complain that positive charges don't come together in 3D, or why are you looking like that? Bad example. But you could still, it does actually happen, and it could be the target here. You could form a hydrophobic cluster where you have something hydrophobic, not hydrophobic, not hydrophobic, not. And when you get closer, when you put them to 3D, you just bring the hydrophobic together, you form a hydrophobic pocket. So there you have something that really happens quite frequently. The positive charges, I maintain my positive charges because there's an existing motif that is exactly that. So how would we find them? How, if we wanted to, if a biologist told us there are these zip codes, and in fact a biologist did, I should put the slide here. So that's the idea of Günter Blobel, who got the Nobel Prize in Medicine for that very idea in 1999, I believe. Now, again, how would we find them? One way of identifying them is we built a profile of similar proteins. So I simply align a bunch of proteins. In this particular case, we have 7. In these 7 proteins, I look for a signal that looks like it's common only in this class. So in this particular case, the proteins share a subcellular localization. Okay? Then I extract this signal, I search for that motif, that signal in the database, and I essentially ask, when I find this, are these proteins of the same subcellular localization? This is a way in which I could identify motifs. And I'm going to show you some of these examples. Now, in the transport mechanism of transporting these zip codes, or using these zip codes for transport in the cell, sorry, there are different types of transport. So some transports are across membranes, some are happening through vesicles, through little lipid pockets, let's call it that way, that travel through the cell. Some are gated, here nuclei, and I'm going to address all of this blue part here, the vesicle transport, because there's something very common to them. And typically they are considered the secretory pathway because many of the proteins that are put outside the cell are processed along this path. This pathway, and there are signal peptides. Now, signal peptides are things, when we look at those, then what we see, so signal peptides are essentially motifs that are recognized by another protein that transports that protein toward the outside of the cell. Okay? When we look at all the signal peptides that are known, that is work, I don't understand, I don't attribute, originally started by Gunnar van Heenje and Henrik Nielsen, I forgot his name, in Søren Brunach's group. Henrik Nielsen was the one who collected all the motifs about 15 years ago and observed the following. First of all, they're typically N-terminal. That means at the beginning of the protein, where the protein starts, the first residue, right? Typically around residues 15 to 30. Most of those are cleaved, meaning that once the protein reaches the extracellular membrane, this part, that is the signal, is actually cut off. So you have something in the ballpark of 30, sometimes more residues that are actually expressed, that are with the protein for some part of its life, but the only purpose of those 30-something residues is to get to the membrane and then outside. Either stick in the membrane or get outside, and then it's cut and digested. Okay? They exist in all shapes of life, so they do it for bacteria and for eukaryotes, and they have a relatively simple, let's call it architecture. So there's this N-terminal region, the beginning of the protein. There's a region that he called H because it's very hydrophobic. There's a C region that has some charges, some polar, uncharged, but it's polar. And then there's the cleavage site, so the point where the enzyme recognizes here cut. Okay? And this is a little bit more detail. What else would you do in order to classify them? So those are words, and you could put these words into an algorithm. Can you? What I put on the whiteboard there, can that define an algorithm? No. There's nothing typically, there's a simple architecture here, often charges, 6 residues, larger than 6. That is an algorithm, larger than 6 residues, but not much more. Rich in hydrophobic residues. Okay, but you have to then define your threshold. What do you call hydrophobic residues? What do you call rich? Does rich mean more than 80% or rich mean more than 50%? Here again, it has polar residues. How many? Which ones do you call? Is uncharged. So how would you do that? How would you fill in the details for the algorithm? Well, one way, of course, would be, I don't have time, I'm just going to invent numbers now, and then by the time, in 15 minutes when the lesson is over, you have the algorithm ready, right? You can do that in the next 15 minutes, inventing your numbers. And then you can simply see whether it works. But you have actually, already in the simple model here, you have too many free parameters to, well, that takes certainly more than 15 minutes to play around with the data set. So, but what could you do that is a little bit better than that? It takes also more than 15 minutes. What would you look at? What type of model could you imagine? I'm not with you at all. So we're back to the profile idea. So essentially what you ought to do is you ought to take all the, this is underneath, this is a collection of all the signals that are known. For the signals that are known, you can sort of align them. You can see exactly what types of sequences do you see. Thus, this you could translate into a profile, you could translate into a hidden Markov model, you could translate into any sort of machine learning, a Bayesian model, so a probabilistic view in which you simply, for instance, say, in this graph here, we see the letter distribution. I'm sorry for the quality, but in principle, the height of the letter is proportional to the information content, the Shannon information of this amino acid. And you do clearly see that at some positions here, and that is aligned with respect to the cleavage side. So there's a minus cleavage side, minus 30. And you see, for instance, that there's clearly a signal for, this is eukaryotes, gram-negative bacteria and gram-positive bacteria. Let's not talk about what that means. But the point is you see signals, okay? This is not flat. Even with these bad plots, you see signals. Not only do you see signals, but you can also, in some sense, see that there are particular signals. So here is an AL. There are Ls all over here. So they are different signals. And these different signals you could simply use to define a Bayesian network, hidden Markov model or something like that, that essentially says, okay, once I have my cleavage site, I go back and I have a certain probability for seeing an L. And according to this probability, I compute the probability for the entire path that this is a signal peptide, right? By the way, here is the length of the signal peptide. They are quite different. I said 30 to 50, 15 to 30 residues. So many of them are much longer. The distribution is different for eukaryote. That's more the 15 to 30 here. And gram-positive bacteria is much longer. But in principle, it's all the same. And the beauty is you can simply use all kinds of machine learning devices to handle the task. Initially, they used neural networks. Then they combined methods, so that's a different story I'll get back later to. And then they used HMMs. HMMs, anybody in the room? I will get back to neural networks briefly later. But anybody in the room knows what the difference is between neural networks and hidden Markov models? No? You have all never had hidden machine learning stuff? Hidden Markov models? So essentially, hidden Markov chains. Okay, I should really, but I did that last semester. Anyway, so you have states, you have transitions between these states, and states in the world of protein typically relates to residues. So you have a certain probability that at one particular position you have an L. Okay? And transition hidden the Markov process is one where the state of this unit is only influenced by what is directly next to it. And since we have a directionality here, what is before it in this particular case, okay? So you have transition probabilities, you have, typically you have an insertion or a skip kind of probability. So you could go to one of 20 residues or to skip no residue. You could go to an end motif step. And for every one of these, say we had 4 amino acids, A, C, D, E. So then from here, and say this would be an A. From the A you have a transition to A, to C, to D, skip, and stop. So in the simplest formulation here you would have 6 probabilities. And these 6 probabilities you essentially learn from data that you have. The data that you have labeled. Once you come up with a new one, you simply compile all the possible paths. And for each path you compute the probability, right? So given these probabilities, you can compute all possible paths and choose the top path, right? Okay. Now, neural network I will talk about in more detail, but the advantage that I mean here is that these types of models, so that's hidden Markov, HMM or Bayesian networks, based on Bayesian inference. The advantage of those tools here is you can put architecture, knowledge, intelligence, expertise into the design of those models. You can do that by biasing, so by inferring how you compute these probabilities. And you can do it by simply introducing different aspects of the architecture. For instance, saying, okay, once there is an A, I know it will always skip 1 or something like that. So I will not even compute the probability to the next one. A will get fast forward to I plus 2 or something like that. This is the kind of thing you can put into the architecture. And in the case of the signal peptides, so you already see here, no, you don't see it. This is a later publication. And it's an advanced method, so advanced means better in many cases, and so HMM do better, and they do better because the problem has a lot of structure. So there's a lot of knowledge that you can put into this problem that will help you, and that's why the HMMs really work much better. Now, there are many other methods that this group developed, and in fact, this group, that is the CBS, it was started from, as I said, from Henrik Nielsen in a collaboration with Gunnar van Heijne. Gunnar van Heijne and Henrik at the point were sitting in Stockholm, but the machine learning part came from the Brunner group in Copenhagen, CBS. And that group essentially pushed a bunch of methods that use a similar idea. Okay? became tremendously successful and was copied many times over. But none of these follow-up methods ever made it to fame. None of them really picked up from what this early method succeeded in. And one of the reasons really is, I believe, that in this community there's a prevalence of old methods. So it takes a long time before a method is established, but once it's established, that is what the community takes. And then to change the community again is difficult. And part of that has to do with a lack of trust in machine learning. Many of the methods that came after were really worse. That's the mistrust has some reasons. Signal peptides. This is N-terminal at the beginning of the sequence, and that leads to a protein being secreted. It's a zip code for secretion, essentially, right? And we have machine learning to learn it. Are there other signals? And yes, there are, and some of them are similar in terms of the way they are structured, and some of them are very different. And that will be part of the lecture on Tuesday. Thanks for your attention. You switch off?